Hello students, welcome to EPG Pathshala. I am Dr. Manjula Sharma from University of Delhi. Today we are going to discuss about the module Spectroscopy from the paper Characterization of Materials 1. After studying this module, we should be able to understand first the principles and instrumentation of UV-Vis IR. First of all, we'll study the principles, then the instrumentation and the instrumental components. Spectroscopy involves investigating the interaction of electromagnetic field with matter. Historically, it originated as the study of wavelength-based dispersion of visible light by a prism. The idea was expanded to include all the interactions involving radiative energy variation with wavelength or frequency. Spectroscopic data is represented as an emission spectrum of wavelength or frequency dependent response. Spectroscopy can be broadly classified into two categories. First, the techniques based on energy transfer between photon and sample, and second, the reflections, refraction, diffraction, dispersion or scattering from the sample, altering the amplitude, the phase angle, the polarization or direction of propagation of the electromagnetic radiation. Over the years, spectroscopy has evolved as a potential tool for experiments and analysis conducted in research laboratories and industries. This technique is essentially considered by analysts as an apparent solution. The objective should also be to use these spectroscopic techniques in control and industrial laboratories and to develop fully recognized spectroscopic techniques. This chapter reviews the interactions of ultraviolet, visible, and infrared radiations with matter. Irrespective of differences in the instrumentation, all spectroscopic techniques have many common attributes. The similarities as well as differences between various spectroscopic techniques have been outlined. Harmonic oscillator is usually employed to model these vibrations, as is shown in figure 1, wherein a chemical bond is depicted as the spring linking between two point masses. Spring tension K denotes the bond strength or molecular force field, and the mass of the atoms or chemical groups is represented by the point masses m1 and m2. The frequency of oscillations nu is expressed as nu is equal to 1 by 2 pi c multiplied by square root of k into m1 plus m2 by m1 m2. Thus, the bond strength decides the vibration frequencies such as triple or double bonds cause higher frequency than the single bonds. Consequently, frequencies are highly sensitive to the type of chemical group, electronegativity of the adjacent atoms or molecules, and the interactions involving hydrogen bonds. If one atom is involved in hydrogen bonding, the bond will be weakened, causing a downshift in the stretching mode frequency. Therefore, the stretching mode frequencies can provide structural details of certain chemical groups such as carbonyl or carboxylic in proteins. This can be achieved by monitoring the formation or disruption in hydrogen bonds. Another factor affecting the vibration frequency is mass of the atoms involved in vibrations. Consequently, the vibration frequency can also be altered by isotope labeling of the atoms involved. Thus, depending upon the type of atoms present, a specific chemical or functional group exhibits vibrational frequencies in particular region. Again, the frequencies in these regions are affected by the environment or the solvents of the chemical group. Thus, a detailed analysis of simplified model compounds in varying environments is required for establishing a clear relationship between the IR mode frequency and structure of the compound. Moreover, from absorption frequencies, the presence or absence of various chemical groups in a chemical structure can be ascertained. Other than the characteristic position of the absorption peaks, their magnitude is concentration dependent. Therefore, both quantitative and qualitative information can be obtained. Students, now let us see the principle. A light beam is passed through an object and wavelength of the light reaching the detector is measured. The measured wavelength provides important information about chemical structure and number of molecules present in the intensity of the measured signal. Thus, both quantitative and qualitative information can be gathered. Information may be obtained as transmittance, absorbance, or reflectance of radiation in 160 to 3500 nanometer wavelength range. The absorption of incident energy promotes electrons to excited states or the antibonding orbitals. For this transfer to occur, photon energy must match the energy needed by electron to be promoted to next higher energy state. The resultant signal is described as interferogram and exhibits such property that each data point in the signal contains information of all IR frequencies coming from source. Consequently, all frequencies are simultaneously measured when the interferogram is recorded. In this way, the interferometer leads to extremely quick measurements. Since a frequency spectrum, that is, intensity versus frequency plot, is required for analyzing the compound, the recorded interferogram cannot be directly interpreted and some methods to decode individual frequencies are necessary. This is achieved by using the well-known mathematical technique called Fourier transformation, and the result of the transformation provides the required spectral information for analysis. Students, now let us see the instrumentation of the FTIR spectrometer. The essential components of an FTIR system are included in Figure 2.2. IR source emits a broad band radiation having various IR wavelengths. Usually, a silicon carbide ceramic is heated around 1550 Kelvin is used as IR source. Rather than a monochromator, a Michelson interferometer is often employed to analyze the infrared beam after passing through sample. The IR radiation passes from the interferometer, which modulates it by performing optical inverse Fourier transform on this radiation. Modulated IR beam is then passed from a gas specimen which absorbs it at different wavelengths due to different molecules present in it. Finally, a detector detects the intensity of IR radiation. A computer digitizes and performs Fourier transform on detected signal to produce IR spectrum of gas sample. Students, now let us see the fundamental components of an FTIR system. First, the source. A glowing black body emits the IR radiation. This beam is passed to an aperture that limits amount of energy incident on sample. Second is the interferometer. Interferometer performs spectral encoding on incoming radiation and exiting signal contains all the IR frequency components. Third is the sample. Beam is either transmitted through or reflected off the sample surface, depending on type of investigations being performed. Sample absorbs energies at frequencies characteristic to it. Fourth component is the detector. The detector measures the interferogram signal. And fifth component is the computer. It performs Fourier transformation to obtain final IR spectrum for examination. The components of optical system in FTIR spectrometer include an interferometer, which has two mirrors, IR source and IR detector, and finally a beam splitter. Beam splitter, a half-silvered mirror, is central to the functioning of the interferometer. When light is incident on it, half of the light is reflected while the remaining half is transmitted. The rotating mirror and stationary mirror of the interferometer both receive each half of these beams. These beams again reach the beam splitter after being reflected from the two mirrors. The beam splitter again splits each half beam into reflected and transmitted half beams. Two output beams result. One travels to the source and the other to the detector. When these two beams come back to the beam splitter, an interference pattern is generated, which is called as interferogram. The displacement of the moving mirror causes difference in path length in the two arms of the interferometer, which in turn varies the interference pattern. The interference pattern is detected by the IR detector as variations in IR energy level and ultimately yields spectral information. If the mirror in interferometer are equidistant from beam splitter, both light beams travel the same distance. When these beams return to beam splitter, each of them is again split and recombined with half of the light from the other arm of the interferometer. The two component beams which are sent to the detector as output are entirely in phase with each other, leading to constructive interference, whereas the two component beams sent to the source are completely out of phase to each other, resulting in destructive interference. Thus, the entire incident light reaches the detector and none reaches the source. On the contrary, if moving mirror is displaced by lambda by 4, where lambda is the wavelength of the incident light, the light from the moving mirror coming to beam splitter is lambda by 2 out of phase with the light from the fixed mirror. This causes destructive interference in the component lights reaching the detector and constructive interference in the components reaching the source. Thus, no light reaches the detector and entire light is sent back to the source. Students, the third instrumental component is the cuvette. Monochromator source is used. Before reaching the sample, light is divided in two parts of similar intensity with a half mirror splitter. One part, or sample beam, travels via the cuvette, having the solution of material to be examined in transparent solvent. Second beam, or reference beam, travels via similar cuvette having only solvent. Reference and sample solution containers have to be transparent towards passing beam. Fourth component is the detector. Detector detects intensity of light transmitted by cuvettes and sends this data to a meter to recorder and display the values. Electronic detectors calculate and compare the intensities of light beams. Several UV-vis spectrophotometers have two detectors, a phototube and a photomultiplier tube, and reference and sample beams are monitored simultaneously. The photomultiplier tube is the extensively used detector in UV-vis instruments. It includes a photoemissive cathode, that is, the electrons are emitted from the cathode when photons strike it. Several dynodes. A dynode emits multiple electrons when one electron strikes it, and an anode. Since the IR source is polychromatic, it emits light with a wide frequency range. Each emitted frequency produces a separate cosine signal, and the resulting interferogram is the sum of all cosine waves produced by each individual IR frequency. In this case, complete constructive interference occurs only when both mirrors are equidistant from the beam splitter, as at this point all waves simultaneously interfere in constructive manner. At all other points, there is only a partial constructive interference. The frequency as well as intensity of each cosine wave present in the interferogram is resolved by Fourier transform. Thus, a plot of intensity as a function of frequency is obtained in the form of a spectrum. Figure 5 shows a hypothetical spectrum having three frequencies with different amplitudes. The middle figure shows the interferograms for these frequencies, and the bottom portion shows the interferogram for the three mixed frequencies. So students, let us summarize what we have learned in this module. We have learned about the principles and instrumentation of the UV-vis IR setup. First, we understood its principles, then we moved over to the instrumentation, and finally, all its instrumental components were discussed. Thank you.